Hello everyone, I am Roshan Kumar, your zoology faculty, and today we are going to learn a very important topic, transport of gases from chapter breathing and exchange of gases. So the topic is transport of gases. So the gases are transported in our body for the purpose of performing respiration. That's why these gases are called as respiratory gases. Respiratory gases, and it consists of oxygen and carbon dioxide. So how are they transported in our body? They are transported in our body with the help of our fluid connective tissue that includes blood and lymph. So occurs by transportation of gas, transport of gas occurs by blood, and a component of blood that is primarily involved in the transportation process is called respiratory pigment, and it is called respiratory pigment. Respiratory pigment includes hemoglobin. Hemoglobin. Okay. So hemoglobin is a protein in our body, which is a type of complex protein. Which is a type of what type of protein? Complex protein. And complex protein means what? Complex protein consists of protein part plus non-protein part. Non-protein part. The protein part consists of globin, and the non-protein part consists of the group heme. It is iron containing non-proteinaceous part of hemoglobin. So let's see how the hemoglobin helps in transport of gases. So we will try to understand it by a simple diagrammatic representation which is far away from its actual structure. We are drawing it only for the purpose of understanding that from which part of hemoglobin these gases are transported. Okay, so let's see. A hemoglobin molecule has 4 globin protein. Okay, so they represent the 4 globin protein. It is what? Globin. As we can see, how many globin protein are there? 1, 2, 3, and 4. We may say it alpha polypeptide, beta polypeptide, alpha polypeptide, and beta polypeptide. Okay, so this globin protein transports its number is 4. It transports carbon dioxide by attaching with its amino terminal. Okay, let's see. What is amino terminal? Actually, we have studied the structure of protein, and any protein is a polypeptide of more than 50 amino acids. Okay, so it is a polypeptide. Polypeptide. Okay, so left end of polypeptide, and this is right end of polypeptide. Okay, so the left end is called N-terminal, or it is called amino terminal. Okay, and the right end is called C or carboxyl terminal. Okay, so then we will say how CO2 is transported by globin protein. The globin protein binds CO2 through its N terminal, through its N terminal. Okay, it binds through its N terminal, and that's why, as it binds with N terminal, we have to incorporate the name amino in its structure or in the complex of hemoglobin and carbon dioxide. So let's see, when hemoglobin binds with CO2, it forms HbCO2, and it is a reversible process. Okay, and this HbCO2 is called carbamino hemoglobin, and I write hemoglobin as simply as Hb. Okay, no doubt. And this amino shows that it binds with the amino terminal of the globin polypeptide or protein. Okay, so here we can see how many globin proteins are there in one hemoglobin molecule, that is number 1, 2, 3 and 4. So one hemoglobin molecule can bind 4 carbon dioxide molecule with its N terminal. Okay, so one hemoglobin carries 4 CO2 molecules. How much? 4 CO2 molecules. No, any doubt? No, thank you. And now let's see, each globin molecule has a heme group in its center, and in each heme group one iron molecule is present in Fe2 plus form. Fe2 plus form. Okay, if it becomes Fe3 plus, then it does not carry anything. It is will called as methemoglobin, and it does not transport oxygen and carbon dioxide. Okay, so let's see, this is a iron containing complex called heme, and its number also is equal to 4, and it carries or transports oxygen. How? So hemoglobin plus oxygen forms HbO2. Okay, and this is called oxyhemoglobin, oxy-HB. Any doubt? No. Okay, then we will again see that how many oxygen molecules can bind with one hemoglobin molecule. So it has HB has 4 heme group, as we can see, and oxygen can bind with each of these heme groups. So one hemoglobin molecule can transport 4 oxygen molecule. And now let's clear one concept, that as we can see, the site of binding of carbon dioxide and oxygen are different with the hemoglobin. So a hemoglobin molecule can simultaneously transport oxygen and carbon dioxide. No doubt. Then let's proceed. That let's think, that is the hemoglobin only molecule that transports all of the oxygen and carbon dioxide? So the answer will be no. And what are the other things, or what are the other forms in which oxygen and carbon dioxide are transported? So to study it in detail, we will study one by one transport of oxygen, and then in next lecture we will study transport of carbon dioxide. Today we are going to study transport of oxygen. Okay, so our next topic will be transport of oxygen. So write the next heading in your copies, transport of oxygen. Okay, so let's see. Transport of oxygen. We will see transport of oxygen. Okay, transport of oxygen, it occurs in 2 forms. Occurs in two ways. The first one is in dissolved state in plasma, as dissolved state in plasma. As we know that plasma is a liquid and 90 to 92% of plasma consists of water, and in that water or in the plasma the oxygen gets dissolved by simply forming any bond. Okay, so in dissolved form only 3% of oxygen is transported. Okay, then the second way in is as hemoglobin, and the rest of the oxygen, that is 97% of the oxygen, is transported in the form of oxyhemoglobin. Do we know that how much hemoglobin molecule does the blood have? So 100 ml blood contains 1.34 gram hemoglobin. That's why, sorry, one minute. As we know, 1 gram hemoglobin carries 1.34 ml oxygen. So, and 100 ml blood has approx 15 gram hemoglobin. Okay, so 100 ml blood carries how much? 15 into 1.34, it approximates 20 ml oxygen. It approximates 20 ml oxygen. Okay, so we can now say that 100 ml oxygenated blood carries about approx 20 ml oxygen. So let's see what happens. We can say that 100 ml oxygenated blood carries how much? So 20 ml oxygen. It is the data which we have, and we will study it in under two conditions. The first one is under normal conditions. Under normal condition, 100 let's see what happens. 100 ml oxygenated blood delivers 5 ml oxygen to tissue. So after delivering 5 ml oxygen to tissues, how much oxygen is left in blood? So that will be approx 15 ml oxygen. That will be approx 15 ml oxygen. Okay, so if the oxygen is delivered to the tissues, and we may assume that CO2 is added into the blood, then the blood will be called as deoxygenated blood. So we can say that 100 ml deoxygenated blood, we write it deoxygenated blood as simply DOB. DOB is not a date of birth, it is deoxygenated blood. Okay, carries approx 15 ml oxygen. So we can say by studying this data that deoxygenated blood is not deprived of oxygen. It also contains oxygen, and but in lesser amount, that is approx how much? 15 ml under normal conditions. Let's see another case during which we are doing some heavy physical works or exercise, that is called under strenuous conditions. Strenuous condition or during exercise. Okay, so the amount of blood in oxygenated blood will remain same, that is 20 ml, and but the amount of blood or the volume of blood delivered to the tissues will be more. And how much? It will be approx 15 ml, because the demand of oxygen by the tissues is increased during exercise or any heavy physical works because of higher rate of respiration in the cells. Okay, so we may say that 100 ml under strenuous conditions, 100 ml oxygenated blood, that is OB, delivers 15 ml oxygen to tissues. Okay, no doubt. So what can we see? Now the volume of or the amount of oxygen in deoxygenated blood will be reduced to, oxygenated blood had 20 ml, it delivered 15 ml, and so the volume of blood left in deoxygenated blood will be 100 ml DOB carries how much blood? Approx 5 ml oxygen. Okay, so this is the transport of oxygen. Now we have understood this, and the next point in transport of oxygen is a very important topic, and we are going to study about that, and that is what? Oxygen dissociation curve. Okay, so let's study oxygen dissociation curve. You know that oxygen dissociation curve is very helpful in studying that how is oxyhemoglobin formed and how it is dissociated at the different sites, that is in lungs and in the tissues of our body. Okay, so we will study it, and it is a very interesting topic, believe me. So the topic is oxygen dissociation curve. Oxygen dissociation curve. Any doubt? No. Okay, so let's see. Oxygen dissociation curve is a graphical representation. It is not a theoretical. It has theories, but we represent the oxygen dissociation curves graphically and explain it theoretically. We will understand it in a simple ways. So let's see, what is it? It is graphical representation between the first one is partial pressure of oxygen. And what do we mean by partial pressure of oxygen? So let's first understand what is partial pressure. Partial pressure is the pressure applied by an individual gas in a mixture of gases. Okay, that we have a mixture of gases, it has a large number of gases or a number of gases, and how much pressure a single individual gas exerts in it. Okay, that pressure is called partial pressure, and it shows, and what literally we will try to understand, what does it show, what partial pressure of oxygen shows in oxygen dissociation curve. So it shows how much oxygen is available to bind with hemoglobin. Okay, it shows O2 available, to bind with hemoglobin. Okay, and between partial pressure of oxygen, and the second one is percentage saturation of hemoglobin with oxygen. Okay, it means how much hemoglobin, how much percentage of hemoglobin of our body has bound with oxygen, already bound with oxygen. Okay, so it shows percentage saturation. Do you know the word of meaning of saturation? It means how much filled space, blank space has been filled. Okay, so here the blank space will be the valencies, or the amount of oxygen can bind with one hemoglobin molecule, and how much hemoglobin molecule, or that, sorry, how much oxygen have been bound with hemoglobin. It shows O2 that has bound with hemoglobin. Okay, O2 that has bound with hemoglobin, no doubt. So it is the meaning of oxygen dissociation curve, and since it is a curve, its shape is an important to remember, as it is very important for our examination point of view. Okay, and so the shape of this curve will be sigmoidal or we call it as S-shaped, or we call it as S-shaped curve. Okay, so now we will study it graphically. Okay, so we will put partial pressure of oxygen on x-axis and percentage saturation of Hb on y-axis, and let's see what are the facts. So on x-axis we will represent partial pressure of oxygen. On x-axis we will represent partial pressure of oxygen. Okay, and it is represented in millimeter of mercury, mmHg. It is represented in mmHg. And on y-axis we will represent percentage saturation of hemoglobin with oxygen. Okay, so let's understand that not 100% hemoglobin is saturated in our body, because the amount of partial pressure required of oxygen required for 100% saturation is approx 140, and in our body it does not reach to that partial pressure. The partial pressure of oxygen does not reach to the point of 140 mmHg. That's why 100% of hemoglobin is not saturated. Only maximum 98% hemoglobin is saturated. Okay, so let's see. The graph will be like this shape, and so if it is 98%, how much? 98%. Okay, so let's see. There will be a point where 50% of hemoglobin will be saturated with oxygen, and for saturation of 50% hemoglobin under normal conditions, how much partial pressure of oxygen is required is approx 25 mmHg. Approx 25 mmHg. So it is 25. Its value is 25. Okay, and this value is called as what? This value is called as P50 value. P50. So let's understand what is P50. So P50 value means percentage या partial pressure of oxygen at which 50% of hemoglobin is saturated. Okay, 50 value is the partial pressure of oxygen at which 50% of hemoglobin is saturated. Understood? Okay, now again we will move to the other side. Okay, and that if it may have two possibilities, this curve has two possibilities. Let's take it as under normal conditions. Under normal condition. Normal. Okay. This have two possibility. Either it will move towards right or it may move towards left by joining the coordinates. Okay. So if it moves towards right, let's study one case. First case, that if the curve moves towards right, okay, we will move it towards right, and let's see what happens. Okay, so we may call it as right shift. We may call it as right shift. Okay, so right shift shows what? Increased affinity with oxygen of hemoglobin or decreased affinity of oxygen with hemoglobin. We may say it by studying the P50 value. Let's see, for saturation of 50% hemoglobin, is the partial pressure of oxygen is higher or lower? It is slightly higher. It is slightly what? It is slightly higher. And higher partial pressure of oxygen required for 50% saturation means decreased affinity. It means decreased affinity. So we may say that P50 value is directly proportional to 1 by affinity of hemoglobin, affinity of hemoglobin with oxygen. Okay, so it is the relation. Here P50 value increased, so affinity of hemoglobin with oxygen decreased. So we may say here that decreased affinity of hemoglobin with oxygen, decreased affinity of hemoglobin with oxygen. If affinity of hemoglobin decreases for oxygen, then what will it leads to? It may form oxyhemoglobin or it will dissociate oxyhemoglobin. So we can say that it leads to dissociation of oxyhemoglobin. It will lead to dissociation of oxyhemoglobin. Okay. Okay. Where we know where hemoglobin is dissociated from oxygen, it occurs at tissue level. Okay. So that oxygen can move into tissue from the blood vessels, no doubt. So it occurs at tissue level. It occurs at tissue level, no doubt. Okay, now we will study what are the conditions at tissue level that are responsible for dissociation. No, so first of all, every cells of the tissue are utilizing oxygen, and as the result of utilization, the partial pressure of oxygen at tissue level decreases. Okay, so we can say factors for right shift are low partial pressure of oxygen, and each cells of the tissue are in performing respiration, and during respiration they release CO2, and because of increased amount of CO2, the partial pressure of CO2 has increased. Higher partial pressure of carbon dioxide. Again, respiration is an exothermic process, and because of the exothermic process, there some amount of energy is released in the form of heat, and due to that heat, the temperature at tissue level is on higher side. So increased temperature. Increased temperature. Again, CO2. What happens? CO2, we know that. What we know? We know that CO2 plus produced combines with water in presence of carbonic anhydrase enzyme. It converts into H2CO3, that is carbonic acid, and it again converts into presence of carbonic anhydrase enzyme into H plus and HCO3 minus. And because of the production of CO2, we may say that the concentration of or the percentage of H plus ion has increased. है ना? So we may say here increased H plus concentration of H plus ion. Increased concentration of H plus ion. This bracket is used to show the concentration, and here we see the concentration of H plus ion has increased. Increased H plus ion means higher acidic nature, and increase in acidic nature leads to decrease in pH. So we may say at tissue level there is lower pH. Okay, and one more product, that is 2,3-BPG concentration is higher. 2,3-BPG, and 2,3-BPG means BPG is what? You may write bisphosphoglycerate. 2,3-bisphosphoglycerate. It is formed during what? Glycolysis. Formed during glycolysis. Okay. So these are the factors responsible for dissociation of oxyhemoglobin at tissue level. Now we will study the another case, and the another case is that this normal curve may shift towards left side. This normal curve may shift towards left side, and we may call it as left shift. We may call it as what? We may call it as left shift. Okay, so left shift shows what? Again it will be answered by the P50 value. Okay, so here we may see that the P50 value has decreased during this time, and decrease in P50 value means increased affinity of hemoglobin with oxygen, means increased affinity of hemoglobin with oxygen, no doubt. And if the affinity of hemoglobin with oxygen increased, it will lead to formation of hemoglobin, oxyhemoglobin, formation of oxyhemoglobin. Okay. Again, where does it occurs? As dissociation occurs at tissue level, so formation of oxyhemoglobin occurs at lungs. Okay, and what are the factors responsible for the left shift? So factors responsible are, number one, here fresh air is inhaled that has higher partial pressure of oxygen. As a result, increased partial pressure of oxygen. Foul air is removed from the alveoli into the atmosphere through expiration. So that's why decreased partial pressure of carbon dioxide. Here the temperature is also on lower side because heat is also released during expiration. So and other factors are also that it is a wet surface and it is also released during expiration. That's why temperature is on lower side, lower temperature. Here this reaction occurs in reverse direction, and it is also performed by carbonic anhydrase enzyme. That's why we may see that concentration of H plus ion has reduced and it has formed CO2 and water, and decrease in H plus ion concentration air means that increase in acidic basic nature, means increase in basic nature, and increase in basic nature means increase in pH value. So we may say that here decrease in H plus ion concentration, and decrease in H plus ion concentration means increase in pH value, and here the 2,3-bisphosphoglycerate and extra product formed during glycolysis is utilized, and its concentration also decreases, that is 2,3-BPG is also decreased. Okay, so it is the oxygen dissociation curve. It is the graph showing oxygen dissociation curve, no doubt. And now we have finished the oxygen dissociation curve. We will compare it with myoglobin, an oxygen storing substance in our muscle cells, and with fetal hemoglobin. Okay, so we are going to compare the hemoglobin and myoglobin, and again the hemoglobin with fetal hemoglobin. Okay, so I guess you all have written this. Now we will move forward. Okay, and one more thing. Okay, let's move. We will study it later in next class. Okay, let's move, and we will move to myoglobin. Okay, it is a protein, and it is also a same type of protein like hemoglobin. The difference is that it has only one globin protein and only one heme group in it, है ना? So let's move and say it is O2 storing protein in muscles. Okay, and it has one heme group, so binds with one O2 molecule and has higher affinity for O2 than hemoglobin. Okay, and because of this higher affinity, O2 leaves hemoglobin and binds with what? Myoglobin. Okay, clear? So let's see the graph again. It shows partial pressure of oxygen, and it shows percentage saturation of hemoglobin with oxygen. Okay, and it's the shape of its graph is shape of oxygen dissociation curve is hyperbola. So let's see, we may take it as It is a shape of hemoglobin. It is sigmoidal shaped, and as we know, the affinity of myoglobin is on higher side, so we will move it towards left or right. Oh yeah, right, you are right. We will move it towards right. So the shape is hyperbola and it is of myoglobin. It is of myoglobin and shape is what? Hyperbola. Hyperbola. Okay. Again, it's all about myoglobin, and now we will study about fetal hemoglobin. Fetal hemoglobin. Fetal hemoglobin. It's similar to, है ना? It means it is alpha 2 and gamma 2. Like adult hemoglobin, it is like adult hemoglobin, that is alpha 2 and beta 2. So, but it has higher affinity for O2 than adult hemoglobin. So, it is like adult hemoglobin, just a difference of only two peptides, that it has gamma 2 and it has beta 2. Its oxygen dissociation curve will also be, shape of oxygen dissociation curve will also be sigmoidal or S-shaped. Okay, sigmoidal or S-shaped. Again, we will study it graphically. It is partial pressure of oxygen in mmHg. It is also in mmHg, and it shows percentage saturation of Hb with O2. Okay. So we will see the adult hemoglobin. So it is adult hemoglobin. Adult hemoglobin, that is alpha 2 and beta 2, and this is fetal hemoglobin. It has higher affinity, and at low partial pressure, fetal hemoglobin carries about 30% more oxygen than adult hemoglobin. Okay, so it has more affinity, so we will shift it towards left. So the fetal hemoglobin, the curve has been made like hyperbola, but actually it's like S-shaped. It is what? Fetal hemoglobin, that is alpha 2 and gamma 2. Okay, so it's our topic for today. It's all for our topic for today's lecture, and we will study transport of carbon dioxide in next lecture. So it's all for today. Thank you. Have a nice day.